guy is doing something which is good for the environment. Even though you might have to spend more for that particular thing, but still you will be looked upon as a person who is who is at least thinking of being environment friendly. As we know, everywhere around us, not many things are happening which look like environment friendly. But what we can do from our side, how we can adopt green technology, not only green nanotechnology, but green way of living. This is what actually makes us socially acceptable. That's why I included this word, socially acceptable. Now coming to the details. So green technology reduces the use of energy and fuel by using less material and renewable inputs wherever possible. Furthermore, nanotechnological products, processes, and applications are expected to contribute significantly to environmental and climatic production by saving raw materials, energy, and water, as well as by reducing greenhouse gases and hazardous waste. Increased energy efficiency, reduced waste and greenhouse gas emission, and decreased consumption of non-renewable raw materials are the main advantages of green nanotechnology. Green nanotechnology offers a great opportunity to stop the adverse effects before they occur. Now here a word I have used is greenhouse gases and their emission, and this links directly to the chlorofluorocarbons, and finally it links up to the ozone layer. But as a hearsay, I don't know whether there is a scientific proof of that or not, that now people have started saying that ozone layer has healed by itself during the COVID times when everybody was back locked in their homes. The ozone layer got sufficient time to heal itself. So I don't know what is the scientific behind that, scientific fact behind that. But yeah, still we have to do our part and we have to reduce the amount of greenhouse gases emission. Whatever we can do, we should try to do. Now, what are the herbal approaches for developing nanoparticles? The activity of herbal medicines depends on the overall function of active components, as all the constituents provide synergistic action and improve the therapeutic value. Each constituent is related to each other and they all play significant roles. And I must say that the activity of a particular plant or a herbal product is not only because of a single phytoconstituent. In most of the cases, many of the phytoconstituents combine together, work in synergy to achieve that particular pharmacological effect for that particular plant. Yeah, there are active constituents which have been attributed to the specific activities and they work in that way. But the plants as a whole, they work together, all the constituents work together. But whenever we write a paper, whenever we compile our results, whenever we write our thesis, or maybe whenever we come up with the conclusion of any project, we are supposed to attribute the activity to a particular phytoconstituent. We need to isolate something, we need to come up with something and attribute the activity to that. Yeah, that might be you can say a requirement, but yeah, it is not that only phytoconstituent that is important. Most of the phytoconstituents work in synergy and attribute the pharmacological effect to the particular plant. Now, coming to the second part, the insoluble character of most of the drugs of herbal origin. It leads to a lower bioavailability, and because of these, systemic release clearance is increased and frequent administration of or a higher dose is required, all of which render the drug a low-class drug for therapeutic use. This happens with most of the herbal drugs. So, to improve this problem, to solve this problem, this herbal approach, the approach for developing nanoparticles is very important, so that we can formulate it as a novel drug delivery system and we can target the specific organ where we want it to deliver the drug. Now, in formulation research, nanotechnology-based dosage forms include the SLNs, the solid lipid nanoparticles, nanospheres, nanocapsules, proliposomes, liposomes, nanoemulsions, phytosomes, transferosomes, etc. They are all advantageous for herbal drug delivery. Now, main important which I will be covering in this talk are liposomes, nanoparticles, microemulsions, phytosomes, and transferosomes or ethosomes. What is the need and why we should adopt the nanotechnology-based dosage forms? First thing is enhanced specificity by drug targeting. Secondly, providing high efficiency and enhanced stability, reducing undesirable effects and toxicity, providing better aesthetic appearance of products. This is very important because you cannot take a plant and as such you can just take a pestle mortar or whatever you want to do, just make a paste out of it and apply it to the skin. It is not aesthetic. It is not acceptable by the body, and even if you try to formulate a product like that, it won't be acceptable in the market as such. So to improve the aesthetic appearance of products, this technology is also very important. Then long-term stability by protecting plant actives from degradation, because when you formulate in novel drug delivery systems, you will use a carrier, you will use a vehicle, you will use some preservatives, and then you will provide a long-term stability to that product, and that will actually help to sustain in the market. Another thing is decrease the allergic potential of herbal substances. So when we decide to come up with a product, a herbal drug to come up in the market, we will eventually try to find out its side effects. If there are any allergic properties, we have to actually work out and how to reduce the allergic potential of that particular drug. So these nanotechnologically based dosage forms, they actually decrease the allergic potential of the herbal drug substance because a very little quantity of the drug is being used and it is directly being targeted to the organ in question. So this is very important in this case. Then improved solubility and bioavailability. That's actually understood that when you will directly target, the bioavailability will definitely be increased. And finally, we can control the drug delivery. Now I'll give some examples of nanoparticles synthesized from plant extracts. First and very important are the gold and silver nanoparticles, copper and copper oxide nanoparticles, palladium and platinum nanoparticles, titanium dioxide and zinc oxide nanoparticles, indium oxide, iron oxide, lead and selenium nanoparticles. You must be wondering, these are all metals, and some of them are heavy metals which can lead to toxicity. But when you formulate all these things into nanoparticles in a controlled environment, in a controlled quantity, and you see that this thing has to be formulated as such that it does not, its deleterious effects are minimized and the beneficial effects are explored. So this is why the scientists have worked out and came up with metal nanoparticles. Most of them, the metals are coming from plant sources. The plants which have this type of metals in them, they are being used and then they are being formulated as nanoparticles, and then these can be used for targeting and for drug delivery. There is an image here showing the nanoparticle of a gold and palladium on an acid-treated carbon support. So you can see how complex is this thing and how much research has to be done to come up with such particular delivery systems. So I will further discuss these things in my coming slides, giving you some examples of nanoparticles from plant sources. Aloe vera, all of you know how important this plant is, and it is a very important plant as far as the cosmetic products are concerned. You can see that aurum and argentum nanoparticles, that is gold and silver nanoparticles, can be prepared from aloe vera, and they having like numerous sizes. The sizes I won't read out, but yeah, they can be they can be in spherical or triangular shape. There are different shapes which actually suit for the delivery purposes. So the nanoparticles which are developed from aloe vera, like Citrullus colocynthis, it gives silver nanoparticles. Curcuma longa, it gives palladium nanoparticles. Curcuma longa, then Magnifera indica, Pisidium guava. These are all the plants which we see in our daily routine, daily life. Not even like Magnifera indica, we all are very familiar with it. Even the clove is present in everybody's home, but we never think that this thing can be used as a nanoparticle. It has palladium inside, and we can use it as a spherical nanoparticle, which can be used for targeting a particular disease or particular health condition. So similar, some of the nanoparticles which can be developed, which are developed coming from eucalyptus, then Magnifera indica, then Pyrus species pear fruit, and then Termidelia cadavera. There are quite of them, and you might go into the details of all this, and you will definitely enrich yourself. That this is a quite interesting topic and quite interesting area where you can actually focus your further research on. Maybe many of you are might be guiding some students to do these things and to do for their thesis and for their PhDs, and these are actually quite useful topics or useful links, useful leads, where you can go inside and you can actually further explore and come up with a lot of fruitful publications. So another one is gold nanoparticles. Why? Because they have attracted substantial attention for their controllable size, shape, and surface properties. Gold nanoparticles have potential applications in areas like biosensors, hyperthermia therapy, antibacterial drugs, genetic engineering, and various delivery platforms for therapeutics. Then coming copper and copper oxide nanoparticles, which are also synthesized from plant extracts. Then titanium dioxide and zinc oxide nanoparticles, which are also important metal oxide nanoparticles that have been synthesized from a number of plant extracts. Gold nanoparticles in particular are actually very important. You might have heard that this is the synthetic way to make it, but when we look back in our ancient text, coming to Ayurveda and coming to the dosage form of Ayurveda, one of the dosage form is Bhasma. Now, what is Bhasma? You might have heard about the gold Bhasma. It is actually very potent. And that is very near to this thing what we have synthesized, the gold nanoparticles. It is just a very fine powder of gold with the bhasma, in which the gold is incorporated, and it is very potent. So these things can be linked back to our ancient texts, but this knowledge is actually not new. This has been actually utilized by our ancestors, by our knowledgeable gurus back there, and they came up with Ayurvedic texts also. So things are linked, and maybe the links for these new things have come from the ancient texts also. So gold nanoparticles, very potent and very important for drug delivery. Now, what are the recent researches which have happened on plant actives and extracts? And just giving some examples, like phytosomes, ginkgo biloba phytosomes, ginseng and green tea phytosomes have got significant attention. They have been got a lot of, you can say, the print media. Lot of print media has involved in these phytosomes of ginkgo biloba, especially. Then liposomes of quercetin, curcumin, and flavonoids for their antioxidant activity. Then nanoparticles of especially of silibene, and then microemulsions of silibene, quercetin, and berberine. You all are familiar with these names. These are quite potent phytochemicals which are directly useful for the treatment of some of the life-threatening diseases. So, incorporating such active constituents into the nanoparticles, or you can say the novel drug delivery systems, into the zones is actually useful, and it has paid rewards for those who have tried to do it, and then now they have become the leads in this particular field of novel drug delivery systems. Now, how this green synthesis of metal nanoparticles take place inside the plant? What happens that these things are actually generated, and how we can generate the nanoparticles? So plant extracts, they are known to contain various bioactives like alkaloids, proteins, phenolic acids, sugars, terpenoids, polyphenols, flavonoids, and many more, which have been found to have an important role in first reducing and then stabilizing the metallic oil. So you can see here that when plant extracts are mixed with a metallic ion source, the biological reduction takes place. And finally, we can get stabilized and capped nanoparticles through various processes. The procedure I'm not discussing here, but the key factors involved in these cases are the pH, concentration, reaction time, which is very important, and temperature, which is the key to the formation of nanoparticles. The shape and size of nanoparticles mainly depend on the variation in composition and concentration of active biomolecules of different plants with an interaction with the aqueous metal ions. More importantly, the nanoparticles synthesized from reducing agents may show general toxicity, engendering serious concern for developing environmentally friendly processes. Now, you should note this thing here, that these things have serious concern for environment. I will come to that in my concluding slides, and what are the problems and how the risk has to be tackled when we are dealing with such type of nanoparticles or novel drug delivery systems, and how to take care while production of such nanoparticles is being taking place. So what should be the strategy for the development of novel drug delivery system for herbal drugs? I am taking an example of curcuma longa, curcumin, which is a very common drug. Everybody knows what is curcumin, and even and most of the developed countries have snatched curcumin that we wanted. Actually, we were thinking about it, and they got the patents on curcumin. So this is actually, then this is not the platform to discuss all that. But yeah, curcumin is very important wonder drug in our kitchen. It is an anti-inflammatory drug, anti-cancer, has got wound healing properties and anti-aging properties, which everybody knows. Then it is one more thing, is photoprotective. Photoprotective means when you apply this thing on your bodies, it will help you to reduce your wrinkles, anti-aging properties, and then it will prevent you from the, you can say, the deleterious effects of the sunlight. Yeah. So coming to what are the problems with curcumin. First thing is poor solubility and poor absorption. Another thing is excess dose required and poor bioavailability. I think this is the universal problem with all of the phytoconstituents. So with curcumin also. Now what shall we do? What should be the hypothesis? The topical administration for, we need to provide a formulation for topical administration for dual effect, including anti-aging and having a cosmetic value also. So what shall we do? And there should be optimum moisture content, so that when we prepare the formulation, it should sustain during the shelf life, and it should actually, you can say, should last long. It should have a good shelf life, that if you prepare today, within 1 week or 1 week or 2 weeks or 3 weeks, the thing is dried, then no point producing such a formulation. So there are variety of things, variety of factors we need to take into consideration. So what shall we do to prepare? Now I'm talking about cream, but here it will be like more specific when we are producing the drug delivery, including the systems like transferosomes, liposomes, ethosomes, or nanoparticles and microparticles. Now, what shall we do is we should target a flexibility in absorption through skin as compared to other drug delivery system, and due to lipid composition, no immunogenic activity because of similarity to cell membrane. Now here they are discussing about the liposomes. Why should we use these zomes? Because they are flexible in absorption because of a lipid barrier, and then due to lipid composition, no immunogenic activity because of similarity to cell membrane. So here one of the major problems is solved here only, that when it is similar to the cell membrane, it will directly pass the cell membrane and reach the target organ without much problem. Now, what should be the methodology? Solvent evaporation techniques, modified handshaking methods, and injection method. These are very common methods. So usually people use the solvent evaporation technique, and injection methods are now widely used also. So this modified handshaking method is, last time I saw, was being used by some of the researchers. They used to prepare the liposomes with by rotating the flasks in their hand. But when I saw them, I was also not very convinced that they are able to prepare or not. But yeah, since it is mentioned, I think it is working. So far so good. So we will focus on the better methods like solvent evaporation and injection methods. Now, what should be the how to optimize the method when we are developing a method for the drug delivery? First thing is proper dose has been entrapped inside or not. Then the release rate and quantity for bioavailability in various media, and then size or range specificity. Coming to the size should be such that it is permeable inside the cell membranes and it is compatible with the lipid bilayer. So this is what first thing is methodology. We need, first thing is optimization, and then how we evaluate once it is prepared. Then for the drug entrapment efficiency, second for the concentration of the composition agent, the first thing which we have used, the agent which we are using to solubilize it. Then we can decide the other things, that what should be the size of our drug delivery system and zeta potential. These are all the things which related to the cell membrane. So when we are developing these things, we have to take care of all these things. Important thing is pH, because a particular drug molecule, a particular phytoconstituent will particularly react, will be active at a particular pH only. And then finally, when we prepare it, we have to do cytotoxicity studies. And cytotoxicity studies, I mean, this one is going to the cell lines in most of the cases, that you have to see whether this thing is cytotoxic on particular cells or not. We have to determine the IC50 for that particular drug molecule, and then we have to find out whether it is in most of the cases MTT assay is being used, and then we determine the IC50 as compared to the standard drugs like doxorubicin or paclitaxel or Taxol. Many of them are available. So usually doxorubicin is used, and then we see if the IC50 is good as compared to the standard. And then when it passes, usually it will work out. But we need to, if not, then we have to rethink and then redesign the formulation, because there will be size issues, there will be concentration issues, and then after a lot of hit and trials, will be able to come up with a product or a molecule which has the cytotoxicity comparable to the standard drugs. Then we have to look for the biodistribution, whether the drug is reaching at the targeted site or not. Whatever you have done till now will go vain if the drug doesn't pass the clinical trials. So ultimately, we have to go for clinical trials. You have to go for clinical trials in preclinical also. Then you have to go for clinical trials, preclinical in animals. And then if you have the facilities for clinical trials, you have to do that also in humans usually. So this is what actually I have discussed in brief, how to develop the NDDS for herbal drugs. So it's quite a complex method, but it pays rewards once the product is in market, once it is actually formulated, and once it is actually free of all the side effects and reaching the target organ without any problem. Then we are successful in development of a herbal drug product with a targeted drug delivery approach. Coming to nanoparticles. Now, in the realm of herbal drug research, which I have seen in like last 15 to 20 years, most of the people, most of the researchers, regardless of the stream, whether they are belonging to uh, formulation or whether they are belonging to like production or the branches of pharmacy like the pharmaceutics or biotechnology or whatever may be the branch, it has become actually irrational now because everybody wants to actually target the plants, herbal drugs, and formulate them in their nanoparticles, because they actually it's good that you utilize the knowledge of that particular novel drug delivery systems and applies to herbal drugs, so that herbal drugs can also be targeted directly as we target the synthetic molecules. So in that context, nanoparticles came into play, and then the nanoparticles and nanoemulsions came into play, and then they developed the colloidal systems. Important and good thing about this is that the particle size vary in size from 10 nanometer to 1000 nanometer. So there is a lot of area, lot of wide actually gap where we can work out according to the size nanonization. You can say nanonization for our particular drug molecule. Again, coming to the advantages of nanoparticles, they are not so much different from the NDDS. That's increase the solubility, enhance the penetration, then reduce medicinal doses as it can be targeted, then improve the efficiency, improve the absorbency of herbal medicines compared with respective normal herbal preparations. This is a simple example I was giving, that we are trying to prepare a cream, but when you prepare novel drug delivery system, when you prepare a ethosome, when you prepare a transferosome, or when you prepare a nanoemulsion, it will be more effective. So this is what it increases the absorbency, and then as compared to the normal herbal preparation. Then another thing is sustained drug release. We can provide it as a pocket which can release the drug in due course of time for longer periods. This is what actually the goodness of NDDS is, that we can provide the drug and it can release itself gradually in due course of time over a period of few weeks also. So this is what actually is the beauty of these delivery systems, that they can be sustained in the body for longer times. Then nanoparticles less than 100 nanometer are commonly used in sunscreens in the form of micronized zinc oxide and titanium oxide to protect skin from UV rays. So many of the people listening here must be using the sunscreens and then using the creams with like saying SPF 50, SPF 100. So what actually is that? That is the property of the cream, or the ability of the cream, ability of that formulation to lock the sun rays from reaching your stratum corneum. So that is what actually is calculated in SPF, sun protection factor. So I'm not going to the details of that SPF, but what I wanted to tell you here is that these are one of the formulations which are utilizing this NDDS products. You can, it's not saying NDDS product, it's an NDDS approach in their formulations. And many of the brands, like one of them is, I don't want to endorse it here. There's one is Banana Boat, and some of them applicable available in Indian markets. Most of them, you might be knowing many of the brands, but some of them which have a higher SPF, definitely they have worked a lot on these to come up with that product. So there are many things going on around our in our surroundings, but sometimes we actually don't think about it. We just observe it, but we never think about it. So there are things which are going around us and which are actually using this type of uh, like, dosage forms or this type of approaches, and it is slowly becoming a part of our life also. Now coming to nanostructured herbal formulations, I'll give some of the examples of the formulations which have gained popularity in due course of time, and this has been list has been prepared on the basis of the research papers which are available on these nanoparticles. Important is triptolide nanoparticles. Active ingredient is triptolide, and it is anti-inflammatory in action, but the method of preparation used for this is emulsification and ultrasound, and it is administered topically. Then nanoparticles of Cuscuta chinensis, they are also hepatoprotective and antioxidant, and they are delivered orally. Then triptolide loaded SLNs, several lipid nanoparticles, they are delivered orally. We saw first one is triptolide nanoparticles, which can be given topically. The second one with triptolide loaded solid lipid nanoparticle, it is given orally. So you can see one molecule or one drug can be incorporated with a change of a dosage form. With a change of the environment, we can give it orally also. Then coming to artemisinin nano capsules, so they are actually very important for anti-malaria and anti-cancer. So they are actually administered intravenously and doing a self-assembly procedure. Okay, they are coming with a you can say a kit where you can assemble yourself and you can actually use it by yourself also. These are some of the things which actually need further research or further research on your part. If you want to know more about these, you can further go into the details of these, and you might get some good topics for further research on in your academic uh, requirements. So another one is the radix salve multirosea nanoparticles, then Taxol nanoparticles, then berberine nanoparticles, silibine nanoparticles, tetrandrine nanoparticles, glyceric acid nanoparticles, and quercetin nanoparticles. As I read all of them, I found out that all of them are very familiar to us. Berberine, we know such a important drug for cancer treatment. Silibine, hepatoprotective. Then glyceric acid is very important, anti-inflammatory and anti-hypertensive, and quercetin for anti-cancer. So all of them are now in the market, or research has been done on most of them using different methods to deliver the drug to the target organ, and different modes of administration also, including intravenous as well as the oral modes also. So now coming to another very famous mode of novel drug delivery, liposome. Initially, when the liposomes came, it was just like a mammoth or something like a thing out of the world. These people are working on liposomes. They are doing research on liposomes. When we used to hear that, we thought there is something special going on, something very big, or the people who are working on these, maybe they are something like out of the world, what they are doing, how they are putting these things into liposomes, what's going on. You cannot see them, how to believe them. There were many questions. But in due course of time, when we understood what these things are and how they are formulated, how they actually help, and then how they are so useful in delivery of the drugs, I really got fascinated with these things actually when I came to know more and more about them. So liposomes, lipid bilayer, chemically reactive. They provide a means of conveniently coupling on a covalent basis. Now, what can be these coupling and coupling tags which can be attached to the lipid bilayer? They can be antibodies, they can be antigens, they can target the cell receptors, nucleic acid probes, etc. But liposomes, what they do, they provide significant versatility in assay formats and detection schemes, and they have diameters ranging from 50 nanometer to 800 nanometers. remember the nanoparticles were from 10 nanometer to 1000 nanometer. They are 50 nanometer to 800 nanometer. They are the ancestors. Nanoparticles came later. So the range became bigger. So the research became bigger. This is what actually is the gradual development in the novel drug delivery systems. Now I will give some examples of the liposomes, which are actually very famous now. The intranasal liposome, especially the quercetin liposomes. Why they are useful? They are useful for their anti-cancer properties and antioxidant properties. They prepared by reverse evaporation technique. Silymarin, I think every novel drug delivery system has utilized silymarin because of its hepatoprotective effects. And here again, the silymarin encapsulated liposomes have been prepared and they were given by the buccal route of administration. Then Artemisia, again it comes here, and then the liposomes have been prepared. Then it is for it is being used for antiviral effects, and it is being administered intravenously. Then paclitaxel liposomes, anti-cancer, prepared by thin film hydration methods, and then entrapment efficiency is 94%. Means 94% of the drug is entrapped inside the liposomes, and the 94% of the drug can be reached directly to the target organ. So such a, you can say, such pinpoint accuracy, almost 94% of the drug can reach the target organ. So this is the beauty of these novel drug delivery systems. Some more examples: curcumin reaching around 88% availability. Then garlicine liposomes, they are targeting the lungs, 90% efficacy. Flavonoids of quercetin and rutin, they are also very, okay, you can say very famous flavonoids from quercetin and rutin. So whoever must have thought of preparing the liposomes is really must be a genius to bring put them into liposomes and give it to and administer intravenously. Because in our herbal drug research, usually when we think about antioxidant activity, we attribute it to flavonoids, and then ultimately we find these two culprits, quercetin and rutin, who are the most of the times responsible for the antioxidant activity in many of the plants. Then usnea acid liposomes are another ones which have been prepared by mechanical dispersion method. They also have attained an 90% entrapment efficiency. Then coming to vogonine and colchicine liposomes. Colchicine, a very important anti-gout drug. It has also been formulated in liposomes. So I really, I know a few other people who are suffering from this type of diseases, and then they really suffer a lot. So sometimes this gout, and then sometimes this lung diseases, sometimes these hepatic problems, they really can trouble people, and if they get this type of dosage forms at the correct time, this type of drugs at the correct time, which can directly target their problematic issues, their problematic diseases, then definitely I think they can actually heal themselves quite faster. So this is what actually is about the liposomes. Now coming to the, you can say, the sister of liposomes, the phytosomes. So they are prepared by complexing polyphenol phytoconstituents in 1:2 or 1:1 ratio with phosphatidylcholine. This is also same. There's a difference is the same, is that phytosome have uh, polyphenol complex with phosphatidylcholine, and then the liposomes have a lipid bilayer. So these are actually very near, but phytosomes have a herbal drug entitled, encapsulated in liposome. Coming to the properties, again, enhanced absorption and bioavailability. They facilitate liver targeting by enhancing the solubility of bile to herbal constituents. As the absorption of chief phytoconstituents is improved, its dose requirement is also reduced, similar to other ones. Then phosphatidylcholine used in the preparation of phytosomes, beside acting as a carrier, also act as a hepatoprotective, thereby giving a synergistic effects. Here, we use phosphatidylcholine in the preparation of phytosomes, and that itself is a hepatoprotective. So unlike liposomes, chemical bonds are formed between phosphatidylcholine and phytoconstituents, so they show a better stability profile. Now coming to the advantages over conventional dosage forms. Phytosomes enhance the bioavailability due to their complex with phospholipids and deliver faster and provide improved absorption in the intestinal tract, hence botanical extracts show better availability as phytosomes. Then phytosomes permit the non-lipophilic botanical extracts to be better absorbed because of the bonding with phosphatidylcholine. Then again, phytosomes can be given in a small quantity and desired results can be obtained. Phytosomes are widely used in cosmetics due to their higher skin penetration and have high lipid profile. Then phytosomes have been used to give liver protective flavonoids because they are easily bioavailable. So more or less the properties, the advantages are similar for most of them, but some of them have some value added effects, you can say value added properties, which make them better than their counterparts or the other drug delivery systems, novel drug delivery system. Give some examples of the phytosomes. Now, first thing is ginkgo biloba phytosomes, and the active ingredients are flavonoids, and then they are prepared by phospholipid complexation method, and route of administration is subcutaneous. Other ones are, again, silibene is here in the list, then ginkgo select phytosomes. Here also the ginkgo, the same as ginkgo biloba, but they have the different properties and the different methods of absorption, different methods of targeting. Then ginseng phytosomes and green tea phytosomes. Now, green tea phytosomes, they are targeting the epicalocatechin. So that increases absorption in the body, and then it acts as a nutraceutical, especially and for antioxidant and subsequently anti-cancer activity. So they are all can be used delivered orally, and then most of them are prepared by phospholipid complexation method. Some more examples here are the grapeseed phytosomes, hawthorn phytosomes, quercetin phytosomes, and curcumin phytosomes. Now, here again we see some familiar names, but these names you will see every time you will come across a novel drug delivery system, because these are the most, you can say, wonderful molecules which can be used to cure a variety of diseases, not only one. These are actually treating a variety of diseases. So they have been the hot topics of the researchers. They have been the hot molecules of the researchers when they taken up by the researchers, and they have been formulated into the novel drug delivery system. In most of the cases, you will find curcumin, you will find quercetin, you will find berberine, you will find flavonoid containing things, because when these are all antioxidants, and then once they are antioxidants, they will act against the reactive oxygen species of the body. This is what actually will affect the research all over the world, because these molecules are very famous for their activity, for their pharmacological activity. Now coming to less, you can say less popular, but yeah, it has been used in the past quite long, emulsions. We all know there are two types of emulsions, oil in water and water in oil. I'm not going to go in details of these because you all know this very well. Now, what is the use? I think there is some disturbance. Okay, so generally emulsion is composed of oil phase, water phase, surfactant and subsurfactants, and appearance is translucent or transparent. We all know about it. So this slide becomes not required. So coming to the classification of emulsions. Okay, ordinary emulsions from 0.1 to 100 micrometer, then microemulsions from 10 to 100 nanometer, and then some microemulsions from 100 to 600 nanometers. So remember the range of nanoparticles was 10 nanometer to 1000 nanometer, and these things are from 100 to 600 nanometers only. So they are the old guys, but yeah, still they are relevant. They have been used to prepare. Mostly the emulsions are used to prepare creams for topical administrations, and it's still quite successful if you are trying to prepare dosage form. Now, what are the applications of emulsions? Now, emulsion distribute in vivo in the targeted manner due to its affinity to the limb. First thing. Okay. Then, in addition, the drug can be sustained release in a long time because the drug is packed in the inner phase and kept off from the direct touch with the body and tissue fluids. So here we can mask the drug with the body and the tissue fluids when we entrap it in the oily phase, or when we entrap in the inner phase of the emulsion. And then secondly, they are non-toxic, non-irritant, and can be easily applied on the skin and mucous membrane. So here also they say that it has to be applied on the skin and mucous membrane, because when you apply on the skin, they are easily absorbed in the skin. You just think of the creams you are applying. They're all emulsions, and some of them are absorbed instantly because they are water based, and then some of them take time because they are on the oily side, oily base. So these are the water and oil and water emulsions. Now coming to the important part is that which are the emulsion herbal formulations which are actually being used in the market also, and research has been done on these particular emulsion formulations. First is the Zidori essential oil with turmeric, then triptolide and docetaxel emulsions are being used, and for their hepatoprotective, anti-cancer and antibacterial properties, anti-inflammatory properties, and anti-cancer properties respectively. The method of preparation can vary depending on the requirement, what we want, which drug we want to release first. So we entrap the drug whether in the oily base or in the water base. And then finally, the route of administration can vary from oral, topical to intravenous. Again, quercetin, silibin, and berberine, they are also entrapped, or you can say utilized in emulsion formulations for their anti-cancer, hepatoprotective, and antioxidant properties. And the activity and the mode of administration can be from oral, intramuscular, and topical. Now here intramuscular is very important because the silibin and emulsion, if it is given intramuscular, it acts as a depot and will release the drug gradually over a period of few weeks or few days. This one can be actually utilized, the one we were talking before, that the drug can behave as a depot, the NDDS can behave as a depot and can release the drug over a course of time. This is what actually we were talking about. Now coming to another one is transferosomes. They can be applied as drug carriers for a range of small molecules, peptides, proteins, and herbal ingredients. So we can say these are a bit more choosy. These are a bit more selective. So you can select special molecules, proteins, and herbal ingredients when you want to target. And transferosomes can be applied as drug carriers. Now they can penetrate the skin, the stratum corneum, and supply the nutrients locally, thereby maintaining the functions and ultimately resulting in the proper maintenance of skin. Most of the transferosome formulations are for the skin products. Now coming to some of the example, capsaicin transferosome, capsaicin coming from the chili. It is an analgesic and a counter irritant, and when they apply topically, it increases the skin penetration. Colchicine transferosomes also increase skin penetration, used for treatment of gout, and they are given IV. Then vincristine from Vinca rosea is given again for its anti-cancer activity. They are given intravenously. Coming to another new formulation, you can say it's quite new in these NDDS, because they have used ethanol, and then you can say they incorporated ethanol into liposome and made an ethosome. So it's structurally similar to liposome, especially suitable as a topical or transdermal administration carrier. And ethosome have a high deformability and entrapment efficiency, can penetrate through the skin completely and improve drug delivery. So since ethanol is easily absorbed by the skin, yeah, definitely it actually will increase the penetration through the skin, and definitely will improve the drug delivery. So it can be used for both hydrophilic and lipophilic drugs. And compared to other liposomes, the physical and chemical properties of ethosomes make the delivery of the drug through the stratum corneum and deeper skin layers efficient and even into the blood circulation. So here we are reaching the blood circulation directly. So in case we want to prepare a novel drug delivery system for a drug which we need to penetrate the skin layers and reach the blood when applying topically, this thing can be used. Ethosomes are a good option. Some of the examples are the metrine ethosome and the ammonium glycyrrhizinate lithosomes. Then both of them apply topically and they have anti-inflammatory effects. So you must be remembering I talked about the risk of this nanotechnology and the novel drug delivery system, especially the nanotechnology approach. What are the risk aspects of these? So the invention of nanotechnology in the field of green technology has raised concerns regarding the nanomaterial impact on health and safety of workers. First thing, when you nanonize a thing, when you reduce the particle size of the thing up to the nano levels, then if the thing is actually like a heavy metal, suppose lead nanoparticles are being prepared and the lead nanoparticles maybe accidentally they come up in the air, they are distributed in the air, some due to some reasons they come up in the air, then they become big biohazard, a safety hazard for the people who are handling them. So this requires a scientific, technological and efforts on part of the government to manage such kinds of risk for the workforce. So here important is the safety of the workforce. The people who are doing this thing, they need their particular safety protocols, and then to manage the risk, how we are going to manage the risk associated with the production of such nanoparticles or the novel drug delivery systems. Detecting genuine risk derived from nanoparticle exposure in the workplace, that is the risk assessment. We must know how much is the risk involved. We must assess how much can be the problem, whether the problem will come from which part, whether it comes from handling part, it comes from storage part, or it is coming from the raw material itself. So these are the things which we need to take care and to plan the control measures of risk management and to finally to communicate the plan. It means like we assess the problem first, find out what are the problems, what are the sources from where the problem is coming out, and then how we are going to solve the problem, minimize the risk, and what shall be the safety protocols, the safety things which we will use so that the workers, the people who are associated with the production of such things are least affected, or they are preserved, or they are saved from these particular exposures. How to assess this risk of exposure? First is hazard identification, hazard characterization, dose response relationships, and assessment of exposure for the different scenarios. Now, hazard identification, where it is coming from. Hazard characterization, how much, what is the character of that thing, how much is the problem, how much is the molecule, how much is the product leaking into the environment, or how much it is on the exposure, how much it is reaching to the people who are using them. So these are all the things which we need to take care. Then those response relationships, that how much is the dose and how much should be the exposure to the people involved there. Then finally to assess the exposure for the different scenarios, whether it is in the research facility, or it is in a manufacturing plant, or it is in a bigger scales, or it's a pilot plant also. So we have to find out whether what is the place where the exposure is taking place and what is the scale of that particular exposure. So unfortunately, the risk assessment process of nanomaterials still suffers from a deficiency of toxicological data for a variety of nanomaterials. So this line in itself clears out that there are not much safety protocols right now in the industry or in this realm which are targeting towards the safety of the workers, and the risk assessment is still not up to the mark. Must be it's an ongoing process and must be during the development of new drug delivery systems. This thing can be actually updated from time to time and can be improved. Then how to manage the risk? That is the first thing is to determine the potential exposure to workers, then identify the potential health hazard, and if possible, eliminate the hazard. Then adoption of green chemistry by substitution with a non-hazard. That's a quite very, you can say very useful thing, that you substitute it with a non-hazardous material. So the problem is solved by itself. Then an introduction of engineering controls, including enclosed systems. So sometimes you can do all the process in a closed environment in such a place where there is no exposure. If it exposed also, it doesn't come out, and the person involved doesn't come in the contact of that particular hazardous chemical, then there is no problem. So risk management, so important thing here is that risk management should be worked out before starting the formulation, or before doing anything, before starting any project. We should assess the risk before, then how we will manage it. If there is any problem, we need to work out before, then we need to proceed to the further stages. Then how, once we know that there are particular risks, then we need to communicate. We need to tell the people involved. This is a crucial part of green nanotechnology, relating to the healthy origination and sustainable development of general public transparency. Everybody should know that this thing has a particular health hazard or hazard, then we need to communicate to the people also. In terms of making available complex technical and health information, risk communication should be made effective in language that should be accessible and understandable to the general population. The most, you can say, the essence of this slide is labeling. When you prepare a particular formulation, the label should clearly state the potential hazards and the risk associated with that particular formulation. And if anything happens, what shall be done to eradicate that particular hazard. So this is what actually is important, is risk communication. So I'll conclude here. Several plant extracts and plant molecules, despite having excellent bioavailability in vitro, demonstrate less or no in vivo actions due to their poor lipid solubility or improper molecular size, or both, resulting in poor absorption and poor bioavailability. This is what we discussed in the first slides itself, that the problem is the bioavailability. So when we want to increase the bioavailability, the important thing is, the solution is novel drug delivery systems which can directly target the organs. Then standardized plant extracts are mainly the polar phytoconstituents like flavonoids, terpenoids, tannins, xanthones, when administered through NDDS systems, so much better absorption profile, which enables them to cross the biological barriers, biological membranes, resulting in enhanced bioavailability. And then several excellent phytoconstituents have been successfully delivered using NDDS. We saw the examples of quercetin, berberine, curcumin. They have actually been successfully delivered using different, all of the NDDS we discussed. These were the common molecules. So the development of novel drug delivery systems for the plant extracts and actives hold sufficient promise, and this is an area which all the researchers should target and should use to develop further formulations, and for their research purposes and for their you can say academic researches also. This is quite a very productive area where you can find so many details, so many useful molecules where you can utilize them and come up with some very fruitful researches which can ultimately benefit the community. So as green technology becomes more commercialized, it will have the potential to become an industry with very strong green credentials. As a general conclusion, I will say that green nanotechnology involves challenging work in the pharmaceutical industry. Ultimately, however, it improves the quality of life, promotes environmental friendly commitments, as well as ethical values in the field of nanotechnology. So the first thing I mentioned when I started is that socially acceptable. This is what actually is that. So when you use these things, you are socially acceptable, then It improves the quality of life. Ultimately, when it is coming from the plants, it is targeting the body or targeting the diseases directly. It definitely will cure you faster, cure the patients faster, disease will eradicate faster, and improve the quality of life of the patient, as well as their ethical values in the field of nanotechnology. So, thanks to all of you for patient listening and giving your precious time. I hope this talk will empower you and enlighten you to further continue the research in this area of green nanotechnology. Thank you.
Thank you so much, sir, for your enriching and insightful lecture. Your valuable time and expertise are deeply appreciated. We now open the floor for questions from participants. Kindly raise your hand or type your queries in chat box. Any question from participants? Participants, please switch on your camera for attendance. Participants can type their queries in chat box. I think question.
I would suggest if all the participants, if anybody have any queries, they can reach out to me on my email. So if they have any questions regarding the presentation, they can reach out to me on my email and I will definitely respond to their queries. We'll try to uh, give the answer in as much detail as possible to satisfy their queries also. So if you are if you have some questions in mind later also, you can come up to me on my email and I will reply to you.
Okay, Dr. Alok, thank you
Okay.
so much. On behalf of organizing team and all the participants of this ICT Atal Faculty Development Program, I would like to extend our heartfelt gratitude to Dr. Alok Natra for his exceptional contribution as a resource speaker today. Dr. Nata's session was not only highly informative, but also deeply inspiring, offering us a valuable insight, but also deeply inspiring and into the latest trend of practical application in novel drug delivery system for especially for herbal drugs. In case of herbal drugs, I think his expertise and engaging presentation have greatly enriched our learning experience and have motivated us to think innovatively about research and development. We are truly grateful for his time, knowledge, and willingness to share his experiences with us. Dr. Nahata's section has set a high benchmark for the set of program and has left a lasting impact on all of us. Thank you, Dr. Alok, for the invaluable contribution.
Thank you. Thank you, ma'am. Thank you.
We look forward to continued collaboration and learning from your expertise in the future also, sir.
Thank you, ma'am. Thank you. Definitely, I will look forward to it. And thank you to your team for organizing it so nicely. So thanks a lot for to all of you. Thanks a lot, and thanks a lot to all the listeners out there. So sometimes the questions come later in the mind. So you're welcome to just communicate with me on email, and you will get a reply from me. Thank you. Okay. Thanks a lot.
Thank you, participants.
Thanks.
Thank you, ma'am, once again.
Okay.
Thank you, everyone, for active participation. We'll take a break of 5 minutes. Then we'll continue. We'll continue with second session. Number of participants. Okay. Dear participants, please note that the attendance is compulsory according to scheme document, है ना. So हम लोग जो है यहाँ पे डायरेक्ट आपकी स्क्रीन से जो भी प्रेजेंट है उससे हम डायरेक्ट अटल एफडीपी के उस पे आपके अपलोड करते हैं. हम बार बार प्रेजेंट और एब्सेंट नहीं कर पाएंगे, इसलिए प्लीज आपसे रिक्वेस्ट है कि आप लोग थोड़ा सा सिंसियरली इसको आप लोग इस पे आपको मेरे को दिखने चाहिए पार्टिसिपेंट. नंबर ऑफ पार्टिसिपेंट जो है मेरे को व्हाट्सएप ग्रुप पे ज्यादा दिखते हैं और यहाँ पे नंबर पार्टिसिपेंट कम दिखते हैं, तो वो चीजें थोड़ी सी मिसमैच हो रही हैं. अटेंडेंस जो है वो सीधे सीधे पोर्टल पे हमको मार्क करनी होती है, अटेंडेंस आपकी फिल करनी होती है. तो रिक्वेस्ट है सभी से कि आप प्लीज थोड़ा सा अपना इस चीज का अटेंडेंस को लेके थोड़ा सा सीरियस रहें.
A very good evening to everyone. Welcome back to the second session of day 5. With immense pleasure, I, Samisha Sharma, on behalf of IES Institute of Pharmacy, IES University, Bhopal, welcome you all to AICT Training and Learning Atal 6-day online faculty development program. It is a great honor and privilege to introduce our eminent speaker, Professor Dr. Vinay Sani, sir. Dr. Vinay Sani, M.Pharm PhD, pharmaceutical scientist, is the Managing Director of Austic Pharma Private Limited, a diagnostic innovation startup incubated at Cine, IIT Bombay, and Director of Tumas Foundation. He previously served as Senior Scientist at the Department of Biosciences and Bioengineering, IIT Bombay, and has gained international research exposure as a visiting scientist at ETS Zurich, Switzerland in 2013 and Fiocruz, Brazil 2024. His research expertise lies in the development of advanced in vitro diagnostic solutions, including rapid ELISA and molecular platforms, CRISPR-based tests, and screening technologies for major public health challenges such as tuberculosis, COVID-19, child malnutrition, neglected tropical disease, leishmanias, filariasis, leptospirosis, sickle cell anemia, Zika, and pox. With a strong translation focus approach, he has successfully brought 9 diagnostic products to the market, along with 8 registered trademarks and 4 patent filed. Under his leadership, Austic Pharma has secured prestigious national and international funding, including the Institute of Eminence grant from IIT Bombay in 2024, BRICS Brazil grant in 2023-25, DST Nidhi for COVID, DST Kavach grant for rapid COVID-19 test, IUSSTF COVID Ignition grant with the University of Florida in 2020, DST-C 2019, and Nidhi Prayas 2019. As co-PI at IIT Bombay, he contributed to major government-funded programs such as India Health Fund, Tata Trust, MDR TB Therapeutics, Imprint India Rapid TB Diagnostic, and DBT Center of Excellence on Child Malnutrition. Dr. Sani has earned significant recognition, including Emerging IVD Company of the Year 2025 by Voice of Healthcare, second runner-up under Glenmark Nutrition Award 2025, top 40 startup selection at Asian Scale Hub, Bali 2024, Asian Entrepreneurship Award finalist, Japan 2023 Startup Award finalist, India UK Conference 2022, runner-up at Global Innovation Forum, Japan 2022, Sarthi Award by Bill and Melinda Gates Foundation, FICCI and ATPC 2022, and prestigious Arohan Social Innovation Award from Infosys Foundation 2020 and conferred by Honorable Shrimati Sudha Murthy and Shri Nandan Nilenkani. Through scientific innovation and entrepreneurial leadership, Dr. Sani continues to advance accessible and impactful diagnostic solutions for global health. With this, we are truly privileged to have him among us today. Please join me in warmly welcoming respected Dr. Vinay Sani sir for his expert talk on empowering pharma faculty by startup creation, entrepreneurial skill, and funding opportunities in transitional research. With great respect, I invite sir to take over the stage. Welcome, sir.
Okay, good evening to all of you. Can you hear me?
Yes, sir.
Can you hear me?
Yes, sir.
Okay, and I hope that my slide is is also visible to all of you.
Yes, sir.
That is visible.
Okay, good. So, okay, thank you so much, Dr. Kavita and Professor Kavita and team from IES Institution, Bhopal, to give this opportunity to me to share this platform and regarding startup innovations, how we can actually, being a pharma person, we can translate our ideas from lab to market. So this way, actually, I believe that people in pharma,
research or biotechnology research, they are having tremendous opportunities to to develop the products and make them available, accessible, and affordable products to the for the benefit of the society. So without delaying of this talk, I am actually going to start this presentation. The title of this presentation is it starts from empowering pharma faculty by startup creation, entrepreneurship skill, and later on we will discuss about the various funding opportunities in translational research through different government and private organizations, specifically for pharma, life science, and biotechnology students, mentors, advisors, and faculty members. So now come to the Startup India Action Plan. First of all, we should understand about the what is Startup India Action Plan. I hope that you are able to see my second slide.
Yes, sir.
Okay. So this plan was laid down by Honorable Prime Minister Sri Narendra Modi sir on 16th of January in 2016. And there was a target to scaling up of a biotech startups ecosystem in India. The target was to establish or incorporate around 2000 startups by 2020. But the target was achieved before this time period. So now India is having more than 1.5 lakh startups in different domains, not only in life science, pharma, biotech, but also in different sectors like digital science, fintech startups in different domains like engineering, mechanical engineering, aeronautics, and different other domains, different allied sciences also. So that so now come to the third slide, which is related to BIREC. BIREC is actually dedicated funding agency for specifically for pharma and biotech startups. So please let me know if you are not able to see any slides. This is our third slide related to BIREC. And you have to confirm from your side. Sometimes it get delayed or server becomes down. So you may not be able to see this slide. Okay. So the BIREC is, so here first two things are important. First one is incubation. Second one is incubating. Incubating is known as company or startup, or incubation is known as a platform like Atal Incubation Center in your institute, in IES institute. So incubation provides a common facility to the different startups so that they can start from their idea to innovations and then do their research, and after validation or preclinical studies, go ahead for next stage, and then for clinical validation or clinical trials introduce after going through the different phases or securing different funding stages. So there are different funding stages under the BIREC for developing the product, not only from idea, but also from proof of concept to prototype development and then validation of that prototype and develop the product and introduce those products into the markets. So there are different types of the categories of the grants which comes under the BIREC. BIREC is the part of Department of Biotechnology. It comes under actually DBT. So in left side, you can see there are different programs, incubation programs, different grants. Grants are related to ideation to early stage, related to ideation to later stage, translational research, equity funding, social innovation, and CSR grants. Right. Likewise, in right side, you can see there are different grants or were announced during a pandemic time period. COVID solutions, under Global Bio India exhibitions were organized by BIREC. Make in India initiatives. First half, technology portals, Grand Challenges India. So Grand Challenges in India, it works with GCI. Grand Challenges India works with PMG, with Bill and Melinda Gates Foundations. So Gates Foundation gives fundings to the BIREC, and then BIREC announces grants for in different domain maybe in neglected tropical disease, maybe in tuberculosis, maybe in COVID solutions, or maybe in futures or next pandemics. So these grants are announced by BIREC. Under National Biopharma Mission, these grants are announced for big companies, for big ideas, for large fundings. And BIREC also supports for different other grants, which we will discuss in next slide. So now you are able to see the bio incubators in 4 slides. Okay, are you able to see?
Yes, sir.
Yeah, I'm sorry to actually to bother you again and again, but I have to confirm. Sometimes we should not stuck in one slide. So bio incubators, nurturing entrepreneurship for scaling up facilities, technologies. It's known as BioNest. So BIREC is nothing but it's Biotechnology Industrial Research Assistance Council. It's a part of Government of India and DBT. So you can see there are different domains. It starts from biosimilar and stem cells in this semicircle or more than half circle, agricultural grants, grants in medical devices and diagnostics, industrial biotechnology and grants related to secondary agriculture. So you can correlate secondary agriculture, guys who are working in or fellows who are working in pharmacognosy, they can work in secondary agriculture or secondary metabolites. Similarly, for pharmaceutics people, they can work in drug delivery and other allied sciences, pharmaceutical analysis or biotechnology. And people who are having interest in biomedical devices and diagnostics, they can work in. Apart from this, nowadays there are multidisciplinary fields. So you can work, if you are, depends on your interest, you can work on AI, ML, artificial intelligence, machine learning, or digital health. So there are so many other grants available in not only with the BIREC, but also with other funding agency or with Startup India program. So now come to the next slide, BioNest. BioNest was launched by BIREC. Its vision was to foster the biotech innovation ecosystem in India, unlike startups in IT sectors. So 10 years back or 15 years back, we must have heard about the IT sector is booming, IT sector is booming. There are so many scopes, opportunities in IT sectors. But nowadays, there are a lot of opportunities and scope also in pharma and biotech startups companies. So the idea was to how we can actually enterprising ideas in the biotech sectors and other allied sciences, including in pharma sectors. So need was to generate the incubation center or establish incubation centers of different kind where they need a landing space to test their ideas and run their operations. So this was the entire mission or vision of the BioNest. And further, to access the high-end instrumentations, as a single startup company cannot actually buy all equipments and instruments. So a common instrumentation facility is available in those incubation centers. And incubation centers not only guide or nourish startups or incubators for facilitate to perform their experiments, but also they give different mentorship. They run different mentorship programs, how to write the grants, how to prepare the pitch, how to prepare the business plan, to prepare a big pitch, good pitch to defend before the juries or reviewers, before the different funding agencies. So they are available in different states, different cities, and they also provide mentorship not only in market research but also in business development, how to prepare these plans under different actually domain. So in case of if you are planning to start your company, then you have to be technically sound. Not only you have to be technically sound, but you have to be sound regarding the how to run management, how to prepare the competitive analysis with the existing products into the market, and how your product would be able to introduce into the market with the help of unique USP, that means unique selling property, value proposition of your products. So these things and competitive analysis, these parameters are considered or giving in the pitch. So pitch is another part I am not going to discuss more about, but here I will be discussing about the entrepreneurship and funding opportunities to the faculty members, students, PhD scholars, and also students, B Pharma, M Pharma, and PhD students. So BioNest program provides support to establish bioincubators either as a standalone entity or as a part of academia. So likewise, in different academic organizations, these opportunities are available. So you can talk to them and start your operations. Okay. Now come to the next slide. Bioincubation saline features. Okay, now it's okay. Saline features. The slide is, we are on slide of saline features for bioincubation.
Yes, sir.
Okay. So this provides incubation space, as I have told you in earlier slide, to startups, connect industry and academia, and enable interactions for efficient exchange of knowledge, facilitate technical and business mentorship, also provide mentorship for IP, how you can protect your idea, how you can file your patent, how you can cover your patent limitations not only in India but also in different parts of the different countries through PCT filing, and technology management regarding later on actually legal and contract aspects, resource mobilizations, and networking platform. Sometimes it happens that in incubation centers all these facilities are not available, so we need to go ahead for the networking, and we have to go ahead for higher studies or higher experimentations, let's say for clinical trials, let's say for toxicity and safety efficacy data in case of the therapeutic part, similarly for diagnostic part. So establish this is an efficient governance model in India through the bio incubation saline features. In left side, you will find that, however, this slide is a bit old, but more than 10,000 proposals were received in the BIREC through different funding agencies, and more than 800 crore funding agencies were committed to different actually incubation centers, and through incubation centers, startups were benefited, or these funds was transferred to the startups, only to the startups who defended and won their presentations or won their awarded with the different grants. So new startups were created, networking and employment was also generated. So you always we have to keep in mind that being a pharma faculty member or pharma professional, not only we are having a, so don't think that you are a job seeker. All of us are job giver also. So don't worry about, we are in good space, we are in healthcare space, we know everything. Even a student from B Pharma is aware about how to develop a tablet, how to prepare a tablet, how to synthesize a tablet, how to manufacture, how to prepare emulsions, different formulations, different pharmaceutical formulations. So they are having fair enough idea comparative to other candidates of other allied sciences. So come to the next slide. This is BioNest scheme. It covers around more than 60 incubators. Those were supported under BIREC program, and funding was given to different incubators. So incubators is nothing but it's only startup companies. A startup tax is giving once your company is incorporated in public domain through MCA website, Ministry of Commerce website, then its incubation is it's a startup tag is started from 0 day to next 10 years after the date of incorporation. So for next 10 years, you can get advantages of different funding agencies, different funding schemes. Not only for male, there are different funding schemes are also available for female founders. So here you can see the focus in technical and business mentoring, world-class infrastructure, and laboratories for startups, entrepreneurship development, addressing unmet needs, accelerating innovations and discoveries. This is most important part, accelerating innovations and discoveries. Legal and IP support, training and programs in emerging technologies, how your technology is globally or domestically competitive comparative to presently existing products, faculty entrepreneurship programs, innovative product technologies, industry academia interactions. So this way, in right side, you will find data which was displayed by BIREC DBT website. So you will get this data also. Now come to the next slide. So now there are different programs, come to the different programs which are being running by BIREC under this for different funding agencies, for funding stages. Incubation, first part is incubation. So suppose if you start your company, then your company will be incorporated with the incubation center. So incubation is known as incubation center, where you will incorporate your incubate your company. Then your company will be considered as is known as incubatee. Then there are different grants from idea to early stage, idea to late stage, then translational research, social innovations, as I have told you in earlier slides. So now come to the next slide. You will get partnerships. So BIREC is also having partnership with different multinational or international funding agencies, whether it is from Australia, France, Gate Foundation, National Biopharma Mission, and other USAID, US agencies also. So now it's come to the next slide. So there are different actually programs also with UK Trade and Investment, Wellcome Trust, Nesta, UNHEI, Tata Trust, Social Alpha. So Social Alpha is known as grant, but it is giving by Tata Trust through another program known as Social Alpha. So sometimes it tie up with BIREC, sometimes it tie up with other funding agencies, maybe other incubation centers, IKP Knowledge Park, and Tata Trust is also running, but another funding agency is known as India Health Mission, IHF. So it gives grant. It's giving it is giving grant for last more than 4 to 5 years. So now come to the left side, you will find equity funding. So equity funding is seed funding. That means a small grant, maybe 50 lakhs to 1 CR. LEAF fund, ACE fund, social innovations. But seed fund is different in different state government funding agencies also. Sometimes seed funding may be 5 lakh rupees, maybe 10 lakh rupees, maybe 20 lakh rupees. So it varies, depends on the availability of fund with different incubation centers or different funding agencies working under different state governments. Now come to the next slide. So these are the bioincubator list of the bioincubators through which you can apply. BIREC doesn't release the grant. Once your fund is released, your startup is selected for final phase, then only after that you will get the funds. But the fund is not directly released to the funding to the startups or incubating. There are certain conditions that your startup should be incubated with incubation center, and through incubation center your fund will be released in different tranche after signing agreement with the incubation center. And next release of the next tranche of your funds depends on completion of deliverables. If you are able to successfully complete the deliverables, then next tranche will be released to you. Otherwise, they will block the grant. So they are competitive and given only after successfully completion of deliverables. Deliverables means activities or target which you have shown during the presentation. So in left side, you will find different incubation centers. This venture center in National Chemical Laboratory, CSIR Lab, Pune, IKP Knowledge Park in Hyderabad, IIT Madras Research Park, C Camp it's in Bangalore, SBTIC it's in Hyderabad, FITT in IIT Delhi, CDB Innovation and Incubation Center SIC at IIT Kanpur, KIT TVI Bhubaneswar, RCB Faridabad, BB Patel Pad Center, Zonal Technology Management Center, GSBTM in Gujarat, STIC in Chennai, KSIDC in Trivandrum, Women Biopark in Chennai. So specifically, female faculty, women faculty should try to approach this Women Biopark in Chennai for further knowing about the different agencies, funding schemes are available with them. then Bangalore Bio Innovation Center, BVC, BITS Pilani, Sign IIT Bombay, Ahmedabad University, and your own Atal Incubation Center. So people who are from IES, no need to worry about this. They can contact with the team of Atal Incubation Center at IES campus and then start writing grants and incubate their companies. Okay. So now you can see in this slide bio incubators. Are you able to see this slide? There are different symbols of bio incubators.
Yes, sir. Yes, sir.
Good. Please, I will bother to you guys again and again, but I have to take care of all these things also. Okay. So these are the different symbols or trademarks or trade word mark or of different bio incubation centers, which I have shown to you in last slide. So now come to the specific grants, which are mainly applicable for you guys. So now come to the most important grant. This is known as big grant, biotechnology ignition grant. This grant is open not only for students, but also for researchers and faculty members. So the purpose of big scheme is to foster generation of idea, and the grant is giving for not only for product development, it's not for developing complete product, it's giving for mainly for if you have any idea and if you have generated some preliminary data, then what you need to do, you have to develop the proof of concept and validate this proof of concept, nothing else extra. Then you will be entitled for this grant, big grant, but you should have some preliminary data as the grant is quite competitive. But grant is also good. Another grant is known as SISTI, Society for Research and Initiatives for Sustainable Technologies and Institutions. So this grant is giving for 15 entrepreneurs, like from GYTI. There is a funding agency in Gujarat. You will get SISTI. So about 15 lakh rupees is giving for 15 entrepreneurial ideas, and 100 young innovations are awarded with 1 lakh grant. To suppose if you are having idea and if you wish to develop something proof of concept, then 1 lakh rupees will be given to you. But if you have already idea and you have developed your proof of concept and then you have to develop prototype, so then you will get 15 lakh rupees. So you have to keep an eye regarding the GYTI. It's also a funding agency which gives grant to different startups and you can apply under this. Third one is EUA. So this EUA grant is given through the BIREC and it is actually quarterly, 6 monthly, or maybe in 1 year it is announced. So you have to keep an eye. So now come to the details about the big grant, biotechnology ignition grant scheme. So this scheme is mainly for scientific entrepreneurs. All of you are scientific entrepreneurs. We are from research institutions, we are from academia, or maybe from startups. So the purpose of this big scheme is to foster generation of ideas, and your ideas should have commercialization potential. Upscale it, validate the proof of concept, and then go ahead for this grant and stimulate enterprise formulation. So this grant is announced twice in a year, 2 times in a year. First one is in 1st January and in 1st July. Now actually, due to certain administrative delays, this grant announced bit late in this year, and it is still open up to 30th of November. So you have opportunities, or you are having 1 more week. You can either actually contact with the concerned incubation center and then talk to them and submit your idea. So this grant value is of around 100,000 US dollar, 100,000 US dollar, but the dollar is so it's up to restricted up to 50 lakh rupees for research projects with commercialization potential, and its duration tenure of this grant is 18 months. So this grant is giving for 18 months. Within the time span or timeline of 18 months, you can develop your prototype and validate prototype and generate data and then submit data to the incubation center and funding agencies. You can, let's say, apply for this grant through different incubation centers which are accessible or eligible for big grant. So earlier, this current big partners were from IKP Knowledge Park, Hyderabad, CCAM Bangalore, FITT New Delhi, IIT, VC and IIT Bombay. Now, few more partners or incubation centers are eligible for big grants. You can contact and you can get the updated list of them in the BIREC website. Now come to the Small Business Innovation Research Initiative. This is known as SIBRI grant. Suppose if you have developed your prototype, already developed your prototype, and you are getting good or exciting results in your prototype. So what you have to do? You have to apply in SIBRI grant, not in big grant. So this SIBRI grant is announced 3 times in a year, 15th February to 30th, then 15th June, then 15th October. And this grant is value is 50 lakh rupees, more than 50 lakh rupees. Now come to the BIPP Biotechnology Industry Partnership Program. This grant is also for developing product as higher scale. Suppose if you have already completed preclinical studies and you have to do phase 1 trials for your product, maybe in Ayurvedic, maybe in pharmacognosy, maybe in some formulation development, maybe in nutraceutical, maybe in cosmetics, then you have to apply for this grant BIPP. So technology readiness level is different for different grants. So for big grants, the technology readiness level is TRL 1 or 2. For SIBRI grant, it's around 3 to 4. And BIPP, it's around 5 to 6. So this grant, like SIBRI grant, this BIPP grant is also announced 3 times in a year. Now come to the important. So these 3 grants, big grants, so all of us are eligible for big grant, but for SIBRI grant and BIPP grants, only startups companies are eligible for this grant. Now come to the important grants, apart from big grant, which are accessible or which academicians are eligible for submitting their projects under these 2 schemes. So there are specific schemes. These are specific schemes for faculties, for academicians, or scientists working in ICMR or CSIR lab. They can apply under this AR. AR means Academic Innovation Research Grant. This is mainly to promote the development of POC, point proof of concept, for a process product by academia with or without industry partner. There is no need to keep industry partner. If you have industry partner, that it will be a positive point. If you are not having, then no need to worry about this. Then another scheme is mainly for academia or faculty schemes is contract research scheme. This is validation of a process or prototype developed by the academicians with the help of industry partner. So here the 100% fund is giving to the academia. But in SIBRI grant, 25% matching fund is required from industry partner. Suppose if your project budget is of around 1 crore or 100 lakh rupees. So BIREC will be giving 75 lakh rupees, but 25 lakh rupees is to be matched given by the industry partner. Similarly for BIPP, suppose if your project is of 1 crore rupees or 100 lakh rupees, then 50 lakh rupees will be given by BIREC, and the rest 50 lakh rupees is to be matched by giving by industry partner as a matching fund. But for AR and CSR grants, 100% fund is giving to the academic partner, faculty members, scientists from different organizations. Now come to the more specific detail about SIBRI grant. So this is giving for early stage research up to 50 lakh rupees. No matching fund is required. Suppose if your budget is 100 lakh, 50 lakh rupees, so there is no need to match the fund. Suppose if your budget is 60 lakh rupees, then 50 lakh rupees will be given by BIREC, and then 10 lakh rupees will be matching fund. Out of this 10 lakh, 5 lakh rupees again will be given by BIREC, and then 5 lakh rupees will be paid by industry partner. So 100 lakh rupees, maximum for 100 lakh rupees, you can write the funds, and its TRL level is 6, early stage validation. As I told you, matching fund for MSME or startup 50% for this, and apart from this, BIREC has added royalty clause also in big grant, but also in 5% royalty clause of annual net sale for VIPP grants. So here you can see a CVD grant, the TRL level was 6, but here you will find in VIPP grant, since this is more advanced grant, so TRL level 7 should be there. That means your product should be in late stage validation up to the commercialization, pre-commercialization stage. Only after that, you will be selected under VIPP grants. So as I have told you earlier in this slide, there is one more grant which is giving under BIREC. It's known as ETA grant, Early Translational Accelerator grant. This is exclusively giving for scientists, faculty members, regular faculty members, and students are not allowed. Sorry, faculty members are not allowed to participate, but faculty members will be participating under this grant with bio incubators or incubation center. So 50% presentation will be given by concerned scientist or academician, and the rest of the presentation will be given by bio incubators. So you will get facility in bio incubation center, and this grant amount is 1 crore to 2 CR. It's not a problem, and this is giving for bio incubators, national research laboratories, government funding laboratories, national institute with demonstrated experience in performing transnational activities in the proposed area, like NIPERS and other organizations. So ETA grant is also specific for, and now it is CCAM, KIT Bhubaneswar, IIT Madras, and some other incubation centers are entitled for ETA grant. So you can contact with the ETA centers and visit their websites for more details. You are still, and this ETA grant is open throughout the year. There is no deadline for this, but specific fund is given to the specific incubation center at a specific time period. So that fund is to be utilized by incubation center at particular stipulated timeline. Now come to the Startup India program. I hope that you are able to see this Startup India slide.
Yes, sir. It is
Yes,
visible.
okay. Yes, sir.
Good. So this Startup India program is actually, everyone knows about Startup India. Startup India was started mainly in 2016, and there are different schemes under DIPP. DIPP is actually certification, and it's given by Startup India or DIPP. So they are having some specific grants of 5 lakh rupees, 10 lakh rupees, 20 lakh rupees, 50 lakh rupees through different centers like Atal incubation centers. They also assist for filing the patent. Suppose if you are going to file domestic patent, Indian patent, then the cost will be reimbursed once your patent is awarded. Similarly, maximum 5 lakh rupees is given by under this Startup India program to file international patent. Once your patent is awarded, then you can claim your money and then go ahead for further research and also innovations and product development. So you have to visit this website. So development, incorporation of a company, it's not a big deal. Anyone can incorporate your company. Hardly you have to spend 15 to 20,000 rupees. You have to contact with your CA or neighboring CA whom you are known. Even your CA who is filing your income tax can incorporate your company. So there are 2 or 3 different types of the companies, like partnership deed company, limited LLP, limited liability partner, private limited companies. But comparative to first two one like LLP or limited liability partnership and um, partner LLP and partners private limited is having more advantages, more weightage, and they are specifically entitled for specific grants and funds. So this way you can go ahead for all these things. Startup India network connect. So Startup India, once your company is incorporated, you have to register your startup with Startup India, and then you will get MSME certificate. And before MSME certificate, you will get DIPP registration certificate. The certificate is giving for 10 years and valid for 10 years. So all startups will be benefited for 10 years and consider as a startup for next 10 years from the date of incorporation of your company. Date of incorporation, that means date of birth of your company. It's like a date of birth of your company. Date of incorporation, it's known as date of incorporation. So from date of incorporation of your company to the next 10 years, it will be considered as a startup, and your startup will leverage, will get so many benefits under this Startup India programs. And you will get a Startup India certificate under DIPP program. So you have to apply under this Startup India website. And different in the upper side, you can see in this tabs about resources, network, schemes and policies, different programs, connect with governments, connect with investors. They also connect with the investors. Okay. Connect with startups, advertising, aeronautics, agriculture, AI, all sectors. So India is actually third largest ecosystem as a startup. So there are a lot of opportunities. Come to the next slide, schemes. What are the different schemes? So the Startup India gives under this Startup India tag, there are different initiatives under Startup India initiatives, DPIIT recognitions for startups. As I have told you in previous slide, there are different government schemes. So apart from BIREC, suppose if you are good in developing devices, then Ministry of Electronics, Ministry of Electronics, Ministry of METI and DETI, these two ministries also give funds from 10 lakhs to 1 crore or 5 crore rupees for different medical diagnostics and devices. Specifically for therapeutic products, BIREC is giving fund. Apart from BIREC, now ICMR is also giving fund for startups. Apart from ICMR, now DST is also giving fund. DST was earlier giving fund also to the startups. And apart from DST, ICMR, BIREC DBT, Department of Pharmaceutical Enterprise, DOP, Department of Pharmaceutical Sciences, which comes under Ministry of Chemical and Fertilizers. So it also gives funds, and you have to check regularly the websites. So nowadays, funding is not a problem, but funding is quite competitive, and your idea should be commercially viable. Commercially viable, that means your idea should have some commercial aspect. No matter whether you will succeed or fail, but you will get opportunity, you will get experience. So no worries about your failures. Even failures are important for our learning process. It's a part of our life. So different programs, partnership program, active programs, participants, international programs. These programs are running by. You can see in the next in this slide, women entrepreneurship in India. So a lot of women entrepreneurship are working very well. They are good award winners. They can execute greatly, and they are working good. So women entrepreneurship, as I am emphasizing, not only for male faculty members, but also for female faculty members to start your entrepreneurship, start your startups companies, and get specific. There are specific grants for women faculty members, women entrepreneurships, in different, specifically in Startup India programs. Female founders. Female founders always, you can see, so at different time periods, Startup India launches female founders, grants for female founders under different schemes. So now come to the next slide. So I have already given an idea about the Startup India programs and all these things. So now I am giving some 5 to 10 minutes regarding what we have done in last 5 years with Incubation Center, and we have successfully graduated. It's known as graduation. Once your startup successfully graduated from Incubation Center, then you have to exit from that space so that other startups will get opportunity to incubate in that space. So fortunately, during COVID time period, we developed our lab, our products. Our first product was rapid COVID antigen test, and it is CDSU and FDA approved. And we donated more than 15,000 tests. Even we donated few tests in Atal Bihari Vajpayee Civil Hospital in Vidisha also, apart from Naga Hospital in Nagaland, Nigris Hospital in Meghalaya, and also in IIT Bombay Hospital. So we are having 8 products into the market, and we are supplying these products to the government hospitals, medical colleges, including AIMS, ISRO hospitals, all military hospitals, ESIC hospitals, and different civil hospitals across all states in India from Kashmir to Kanyakumari. We have recently launched 2 nutraceuticals into the market. One is exclusively for overall well-being for female. It is known as Ostic. And another one is for overall well-being for male, ostestosterone. So they are for overall well-being. That means immunity boosting, is for good skin, good hair growth, good reproductive health, and exclusively this male product is exclusively for reducing cholesterol, giving energy. It is very good product. People who are doing gym, and both products are good for fatigue, letharginess, and for activeness. So this way, we have introduced as during introductory time period, we are also running a novice foundation. This is known as Thomas Foundation for tuberculosis, malnutrition, and AIDS. So we do social innovations, and we were awarded by Honorable Sudha Murthy Madam just before pandemic in 2020 for developing our TB diagnostic test kits in 2020. We were awarded through Startup Award, Sarthi Award by Gate Foundations for our good work and donation of 15,000 tests of around 80 lakh rupees during that time period during pandemic. These are the interactions with Honorable Prime Minister Modi in 2018, HRD Minister, the then HRD Minister. We completed different projects through our foundation also in Uttar Pradesh, Jhansi with child malnutrition. And successfully we completed screening of 10,000 children for their malnutrition status in Mawal and Mulsi area in Pune with IIT Bombay and Department of Biotechnology with our Northeast partners, Arunachal Pradesh and also Nigris Medical College in Meghalaya. So these are the interactions with former US Ambassador during Indo-US Science and Technology Forum funds during COVID time period, Eric Garcetti. These are the presentations in NIPER, CII, and exhibitions, interactions with DCGI, Honorable DCGI, Mr. Chang Ji-bok, Ambassador of South Korea in CII meeting in Pune. In left side, you will find Director NIPER, sir. And then during my talk, we were selected under this Asian India Scale Up program to present this work last year in Indonesia also. 40 startups were selected under this program through DST and IIT Kanpur. So we presented our product in ASEAN countries. 8 to 10 countries participated in Bali, Indonesia. We were actually awarded this year in IBD company or emerging IBD company by Voice of Health. And so you can transfer your technology to the startups also. So one technology for swine transmitted helminth infection was transferred to us, And these are the pictures in right side with policeman for our nutraceutical product during Tech Fest in IIT Bombay. Recently, last week, I completed an important trip in with India UK MedTech roundtable discussion, which was organized in London. And so we got an opportunity to interact with Indian High Commissioner in High Commission of India at London, and also got an opportunity to interact with health ministers, Minister of Parliament in UK Parliament on 13th of November last week. So this way, these are the crisp presentation. This is crisp presentation, and now I am open for question and answers, and would be happy to resolve your queries if you are if you have any question without any hesitation. As always, I consider like students, so don't worry about. No need to worry about. Don't hesitate. Do well. You guys are already having potential, knowledge, and power to get it converted into translational research and product development. And I believe that pharma people are having tremendous energy, knowledge, and my request you guys to translate your ideas for providing innovative products to the underprivileged population and do some social innovations. And this way, you will lead the foundation, make the foundations of India under indigenous development of products under a Startup India, and make a sustainable, affordable, and provide affordable solutions in healthcare sector. Thank you so much for your time and attention. Now we are open for question and answers.
Thank you, sir, for an excellent and thought-provoking lecture. We now invite questions from participants. Kindly post your queries in the chat box or unmute yourself.
Hello, sir.
Yes, please.
Sir, good evening, sir.
Good evening.
Sir, the kind of project I'm applying.
We are in Big Run.
Sorry, sir.
Where you are applying?
No, sir. Actually, for the any kind of project, if I wanted to apply,
Okay.
will it be major content should be from the drug-based content or the technology-based content? Which one is good for me to get the approval, sir?
So if it is a new drug, then it will be considered the innovations, right?
Yes, sir.
If
But
it is a new
it
drug,
requires
it will
some
be
kind of background setup also. So, is it good for me to apply for the scheme with the setup requirement?
no setup requirement for requirement of setup. It's actually different grant. So setup requirement is actually giving directly. You will not having access for setup funds for setup requirement. But if your company is comes under MSME or startups, and if you have successfully completed and deliver some products into the market, then your company or startup will be entitled for big grants. Let's say for 5 CR or 10 CR, 10 crore rupees or 5 CR, 5 crore rupees from different under different funding schemes. But there will be matching fund, and so nowadays actually government is not giving entire fund in the form of grants. There is part of the grant, let's say 50% will be grant, 50% will be loan.
Okay, sir. Okay, sir. Thank you, sir.
So it's better to apply not for product development, not for setup development. However, you can develop your own setup, small scale, small setup with the fund, available fund of the fund which will be released. So you can allocate fund for different machineries, equipment, and instruments. But there are certain restrictions. You cannot give more than 20% fund for non-recurring cost and more than 10% fund for manpower cost, human resource.
Okay, sir. Thank you, sir. Thank you very much.
So are you able to, am I was
Yeah, very clear, sir.
I'm
Thank you,
asking
sir.
whether queries have been resolved or not?
Yeah, it's clear, sir. It's clear, sir. Thank
Okay.
you, sir.
Any other guy?
Sir, hello, sir.
Yes, please. I can hear you, Mr. Perdoman.
Yes, sir.
Yeah. Please tell me.
Uh, Sir, my question is that which product is easy to come in market, Ayurvedic product or sir synthetic based product?
Ayurvedic, obviously, Ayurvedic product, natural products, right?
Okay, sir. Okay, sir.
Natural products, nutraceuticals, Ayurvedic and
Okay, sir.
nutraceuticals, and cosmetics.
Okay, sir. Okay, sir.
So always you should start from simple things.
Okay, sir.
This way, once you introduce simple product into the market, I am not saying all Ayurvedic, cosmetic, and nutraceuticals are simple comes under simple product category of simple products, but they are easier to exit into the market comparative to complicated new molecules or
Yes,
new
sir.
formulations.
Yes,
Right?
sir. Yes, sir.
Go ahead.
Thank you, sir.
All the best.
Thank you, sir.
Welcome. Any other question? I cannot hear. I could not hear anything else from women founders, women entrepreneurs, women faculty members.
Okay.
It seems that it's a dinner time for all of them.
Okay, Dr. Vinay, thank you so much. On behalf of coordination team, I would like to express our sincere gratitude to Dr. Vinay Saini for his invaluable contribution as the resource speaker on the day 5 of Atal FDP. We deeply appreciate your time, knowledge, and inspiring insight that have greatly benefited all participants. Thank you, Dr. Vinay, for your enlightening session and your ongoing contribution to pharmaceutical innovations. Really, it was an honor to have with you us. Thank you so much, Vinay.
Okay, pleasure meeting. Okay, should I leave? Thank you so much.
Participants, please switch on your camera for attendance purpose.
So do we leave or is there any other formality?
With this, we conclude the day 5 of Atal FDP. We extend heartfelt gratitude to AICT, Atal Academy, our respected dignitaries, resource person, organizing committee members, and all participants for their support and engagement. We look forward to your enthusiastic participation in the upcoming sessions. Thank you, everyone, and have a wonderful learning experience ahead.
So thank you so much for your time and attention. Wish you all the best.
Thank you, sir.
We all can leave, madam.
Yes.